This is a dynamic molecular simulation showing a protein binding sequence in constant motion. Simulations like this are driving modern drug discovery, using vast biological datasets to mathematically predict how potential cancer and antiviral treatments will interact with human cells. But predicting these interactions generates an enormous amount of raw data. To actually understand what the software is predicting, researchers need robust visualization tools to translate those numbers into a format a human scientist can analyze. By the end of this tutorial, you will know exactly how to pull raw information from the protein databank, clean it up into a docking-ready file, and export a clear, publication-ready 2D diagram of your results. Mastering this workflow translates computational data into visual models. It allows you to take complex AI-generated predictions and turn them into actionable insights that can be shared with your research team. Step 1 is getting our target. Open your Discovery Studio Visualizer. Head to the search menu, type in PDB-ID 204, and hit Download. Once the file loads, open the hierarchy view. To make the raw structure easier to see, take a moment to adjust your workspace background color until the molecule stands out clearly. Take a look at the newly loaded protein structure. You will notice it is surrounded by an extensive field of tiny red dots. These are water molecules. Looking at this side-by-side comparison, we need to move from the raw state on the left to the cleanly stripped protein on the right. In your hierarchy panel, select all those water molecules and unnecessary heteroatoms and delete them. While you are clearing out the noise, be extremely careful. Make sure you do not accidentally delete the native benzimidazole ligand, as we need that bound structure for the next step. Starting with a rigorously cleaned protein structure, free of artifact water molecules, is a critical foundational step. This cleanup prevents the software from generating false positives during your future docking simulations. Now that the structure is clean, locate and select that native ligand we carefully preserved in the last step. Here in the interface, click the Define and Edit Binding Site button. This generates a translucent binding sphere directly around your current selection. This next step is mandatory. Right-click the newly generated sphere to open its attributes panel. This diagram maps the XYZ coordinate axes intersecting at the center of your 3D binding sphere. Write down these exact coordinates from your attributes panel. They dictate the spatial grid for future docking algorithms. With these boundaries recorded, safely delete the native ligand. Next, navigate over to the Chemistry tab, select hydrogen, and execute the command to add polar hydrogens to the entire structure. To visually confirm the structure is fully prepped, change your primary display style to the ribbon form. By accurately defining these binding pocket coordinates and adding the necessary polar hydrogens, you provide the geometric and chemical parameters external software needs to calculate the most probable binding poses. Let's move forward to the visualization phase. For this step, ensure you have both your protein and a docked small molecule loaded together in the workspace. Open the View Interaction panel and click the command to Show 2D Diagram. This key categorizes chemical bonds by line color, a logic the 2D map uses to depict distinct molecular relationships. Notice the green lines. These isolate conventional hydrogen bonds. Then we have the orange lines, which indicate pi-anion interactions within the structure. While the data is accurate, the software has a primary failure point here. The default text labels generated on this 2D diagram are almost always too small to read comfortably. Looking at this split-screen layout, we want our results to match the readable version on the right. Right-click the background, access display size, and increase your label font size to a 3 or a 4. Finally, initiate the Save As procedure. When prompted for the export settings, make sure you set a high pixel ratio to ensure the image stays sharp. Adjusting these specific display parameters is what transforms raw, cluttered software outputs into crisp, publication-ready assets fit for scientific journals. Go ahead and open your finalized image file. Check to verify that you have a sharp, high-resolution rendering of your protein-ligand interactions. Creating clear, legible visualizations like this is a daily requirement for teams working in computational chemistry and biotech research and development. This is a dynamic model of a protein binding to a target. When researchers have clear visuals of these mechanisms, it accelerates the decision-making process for engineering new therapeutic proteins and understanding drug resistance. Mastering this specific preparation and visualization workflow allows you to effectively communicate your structural findings, ensuring your data is formatted and ready for formal presentation.